start in a moment. I think we've got a few more people to let in the door, so we'll just wait another minute or so and then we'll get going. Well, I think we're pretty close to the numbers we were expecting, so I think we'll get going. You can move the slides along, Kariana, to the next, to the beginning, if you would. There we go. Great. All right. Well, good evening, everyone. Let's get started with our water quality training session. My name is Deborah Martin Downs. I'm the chair of the Environment Committee, and I'm going to be leading you through the program tonight. I am a second generation cottager on Lake Muskoka, and I'm a member of the East Bay Water Quality Testing Team as well. So welcome to all our water quality sampling volunteers, and a special welcome to those of you who are new to the program. I see some of the names of you that I've been corresponding with, so thank you for coming today. And you may be new to the MLA as well, so welcome to the MLA. Thank you all for putting your hand up for Muskoka. It's a very valuable thing that we do here, and the members appreciate it so much. Next slide. So this is our agenda for the training. We're first going to talk about why we sample, followed by some of the results from our 2024 year, and then we will delve into the training portion. We have a lot of material to get through, so I will endeavor to do so as quickly as we can, but because we have new samplers joining us today, I want to make sure you're feeling confident to hit the water as well. So we will have time for questions after the major segments. If you want to have a question, ask a question of anyone on the panel, please put it in the Q&A. That button is found at the bottom of your screen. Unfortunately, the Zoom webinar format doesn't allow you to speak or to be seen, so we will have to rely on that method at this point. Next slide. Before we proceed, let me introduce our presenters today. And once again, I'm Deborah Martin Downs. I'm MLA Director and Chair of the Environment Committee. I'm joined by Louise Craig, lead for the Beaumaris north side of Lake Muskoka area sampling team, and Jen Getson, lead of the Lake Muskoka East Bay team, stars of our training videos, you will notice. And last but not least, Kariana Costa, our MLA staff person with responsibilities for the program. And you've all probably had a conversation with her at some point in time or picked up your bins from her. So, and of course, we all thank you as our volunteers. You're the most important part of our team, and we couldn't do any of this without you. Next slide. So I want to start with an overview of the program, and what we do is a bit of context for the training. Next. So when we look back at member issues over the years, even in 1894, when the MLA was founded, water quality has always been top of mind for Muskoka waterfront property owners. There's nothing worse than having a lake in front of you that's unsafe for swimming or drinking. Fast forward to 2021, when we were doing our strategic plan, we asked our members what they valued about Muskoka most, and 93% said they valued water quality great for swimming. Further, 88% said they were worried about the degradation of water quality in the future, and that was followed closely by boat noise. And those in the graphs, those are the top. You can't read the small lettering, but the green bar on the very top is the answer there. And in our more recent MLA survey, similar results to the green bar were also found. Next. And then we asked what the MLA should be spending more time on. And no surprise, actively promoting protection of the environment and water quality ranked as the top choice, shown in the green bar, followed closely by actively promoting responsible development of the waterfront. And these two are very much linked. for those of you who may not be aware, the MLA has a very active government and land use committee who engage on many files across the 4 municipalities we work in. We advocate for responsible development, among other issues. The Environment Committee also has members sitting on it to ensure that we coordinate our responses and that we understand the implications of some of the work that's happening out in Muskoka. Next slide. So if you've been following some of the news, local news in the past year, you'll have heard the term watershed and watershed management mentioned a lot. And we're fortunate to have the Muskoka Watershed Council active in Muskoka, and they actively educate our elected officials on the benefits of watershed management, and they're advocating for the development of a watershed plan. You'll be happy to know I'm not going to launch into Watershed Management 101 talk, but I want to provide you some key concepts for you as you understand this. So the biggest concept is you can't take the watershed out of the water. And that means its rock, its soil, its vegetation, the urban areas, its topography, and its climate all influence the quality and quantity of water. The watershed of the Muskoka Lakes is huge. The headwaters of the river arise in the western slopes of Algonquin Park, and then they flow for approximately 210 kilometers toward Georgian Bay. The watershed measures over 62 kilometers at its widest point and approximately 120 kilometers long, and encompassing an area of 5,100 square kilometers. So massive watershed. Inside it, we have 42 dams and 36 lakes connected by over 170 kilometers of river. And making it like really the most complex, one of the most complex watersheds in the province. And while we're very fortunate that much of the watershed remains natural, there's land development, resource extraction, and natural factors all work in the watershed that can affect the quality and quantity of our waters. So we have to look to the whole watershed to understand what might be happening now or into the future in the lakes. And that's what a watershed plan can help us do, is predict that future. So while changes at the watershed scale affect the lakes, smaller scale changes can also be very important. Our members have rightly made the distinction between, or the connection between development and other changes on the landscape and their effects on the water. So every land use change along the shoreline has the potential to affect water quality, albeit most of the time localized. Even when we've done our best to keep our properties natural, we all have an effect. I have an effect too. We all have cleared part of the land. We all have a septic system. And perhaps you too have a family member who just has to wash their hair in the lake. But the conditions at each site may be unique. And whether they're wave exposed or in a shallow bay, it's important to measure them as well. It's hard to see the impact of those changes, though, on the whole system. And there's a term called cumulative effects, and that's really what we're trying to measure when we go to the deep water sites to basically see the cumulative effect of all those changes on the lake. Next. So this figure provides just a simplified depiction of the aquatic ecosystem of our lakes. Light and nutrients control the productivity of a lake. All plants need light to grow, but equally important, they need nutrients to grow, just as people and plants in our gardens, the same thing. And the nutrient that controls plant growth in our lakes is phosphorus. And phosphorus occurs naturally because it is in every organism and in every plant. But it can be added by things like fertilizers. And recently, goose poop has become a bigger thing. Could be added through poorly maintained or failing septic systems. And like all things, too much can begin to make changes to the ecosystem. Now, I don't claim to be an expert in all aspects of this, but we measure phosphorus because most of Muskoka lakes are naturally low in it, resulting in low productivity. And in scientific terms, we call that an oligotrophic lake. I'll mention this in a bit more. So when we have too much phosphorus, we'll see more algal growth. And if our system is in good shape, then the zooplankton, the ones in the next level up, eat the algae, and they're in turn eaten by little fish, and little fish are in turn eaten by big fish, and some of the big fish are eaten, or the little fish are eaten by birds. So that's the chain of events. But if we break some of that chain, or if the chain can't respond, then we will have changes to the lakes and the water quality. Algal blooms can result by having too much nutrient, and some of our lakes appear to have that problem. But our lakes are not as likely to have that nutrient increase, creating blue-green algal bloom problems as much as climate change-induced impacts, such as reduced wind and increased water temperature. Next. So hence, when we sample, we're trying to capture different effects from the larger scale down to the smaller scale. So our deep water sites are typically more consistent. The change as the water flows through the lake and mixing with wind and wave action. And here's where we monitor phosphorus concentrations at our deep water sites, and mainly for long-term trends and that complete mixing of the lake. We monitor water clarity there with our Secchi dips as well, and water temperature to help explain unexpected results and help determine trends over time. And for some sites, we'll be doing chloride, which I'll mention in a bit more. We also monitor nearshore, and in this picture, it's shown as the littoral zone, and that's to capture the effects of creeks, of runoff, of groundwater interaction, and including things like wave and boat impacts as well. So here is where we would undertake any bacterial sampling that we do. Not all sites do that. And those are largely in areas where we would expect to have more elevated bacterial levels particularly around recreational areas that might affect recreational areas. And we also monitor phosphorus concentrations at nearshore sites to identify sources of phosphorus, such as septics and outfalls and oozes or fertilizers from golf courses or other such things. So we sample from May 24th weekend. I know it's a hard time with many opening a cottage as we do, but we try to do it at this time to capture a time when the whole lake is mixed up, and that's known as spring turnover. So what do I mean by that? It means that the lake turning over is a seasonal mixing of the entire water column. So for many lakes that are deeper than about 20 feet, during the summer, we get thermally stratified layers of water. And the layers prevent the lake from mixing and aerating. So on the chart on the, at least it's my left, I'm not sure what left is going to be. It has 3 different colors of blue in the summer chart. It shows you've got warmer temperatures at the top, and then a middle area in the middle, the thermocline, is where the temperature starts to rapidly drop. And then it stays at a fairly consistent temperature at the depths. And any of you who are divers know the deeper you go, the colder it gets. And in the bottom, it typically stays at about 4 degrees Celsius all year long. But then when fall comes along, the temperature of the water begins to cool, and eventually the layering that begins to weaken the separation of the layers, and allowing the lakes to mix when the temperatures equalize. So as the temperature, the density of the water changes to be one type of density, the water can mix. And that means the water from the bottom of the lake can come to the top, and the water from the top sinks to the bottom. And that process allows oxygen to be replenished and nutrients to be distributed throughout the lake column. And this temperature-driven process of lake turnover also allows aquatic life to inhabit the entire lake, because oxygen becomes more available at the bottom. And without that, our lakes wouldn't be as vibrant and dynamic as we are today. This is especially important for lake trout, and some of you will have lake trout lakes, and oxygen concentration of those lakes is super important because they like their lake cold and they like it oil oxygenated, and they can't live in the warm water where the oxygen would be. And then as the season progresses, we just sample the top of the lake, or where the water is trapped. And in the near shore, this process doesn't happen. So it's in the open lake water where we're trying to see that effect during the year. Next. So moving from the theoretical to the MLA program, this is the area within which the MLA water quality program operates. So Bracebridge, the lower part of Seguin Township that drains to Rosso and Joseph, Gravenhurst, and the township of Muskoka Lakes. The sampling on the 3 big lakes and connecting rivers is supported by the MLA membership, and other lake associations join the MLA program using our methods, our lab services, and the analysis of the results by our consultant. And doing so can achieve a water quality program for their lake at a much lower cost. So the citizen science, as our program is known, became all the rage in the early 2000s. And the MLA started our current program in 2002, although for years before that, there were other programs that were offered. But this is how we've been running it now for over the past 22 years, which is a wonderful record to have now to see long-term changes. The Water Quality Initiative has become one of the MLA's most significant programs, and thanks to our, I'll call it a small army because there's almost 100 of you doing this water sampling at 54 areas and 17 different lakes and rivers, volunteering your time and your boats through the lake, through the lakes of Muskoka to collect this information. The samples, when they're collected, are sent to an accredited laboratory, and the results are analyzed and reported by our environmental consultant, Hutchison Environmental Sciences, founded by Neil Hutchison, who designed the program for the MLA at the beginning in 2002. The key objective for our program, though, is to determine if the amount, nature, and activities associated with shoreline development are altering key water quality indicators. And shortly, you're going to learn how to measure those things. So, okay, be patient. We have some areas to document baseline conditions. Where we do ours in East Bay, there's very little development, and so it sort of tells us whether how the natural condition of the water will be. And then there's many other nearshore sites that are typically positioned near golf courses, resorts, marinas, towns, landfills, or aggregate operations. And there, of course, I mentioned earlier, we sample for phosphorus and water clarity and bacteria in the near shore to identify those issues. Recently, we've we've added chloride, as the science is telling us that road salt can impact our lakes at very low levels. In southern Ontario, higher levels are tolerated. And then we also document harmful blue-green algae occurrences as identified by the Ministry of the Environment, Conservation and Parks, or the Simcoe Muskoka District Health Unit. There's a lot of other players in the water quality and environmental monitoring space in Muskoka, and some of you will be doing samples for the Lake Partner Program, and that is run by the Federation of Ontario Cottagers Associations for the Ministry of the Environment. Some of you may have recently received a letter from FOCA asking you to send a message to the ministry saying get on with it and make sure the funding is in place. Thank you if you have done so, and if you are one of the Lake Partner Program sites. One of our sites near Windermere, we monitor for algae every 2 weeks, and that's part of the Muskoka Watershed Council's algal monitoring program. The district also samples all the lakes in their area on a rotation roughly every 3 years, but they only do deep water sites, which is fine for what they're doing. And then the work that MLA does with the nearshore sites really augments that kind of work that they do. And then, as I mentioned, you've been hearing a lot about road salt, and this is largely the work of the Friends of Muskoka Watershed, who have their Salty Muskoka project and who are looking at the science of salt effects in Muskoka and trying to get people to reduce their uses and to look at the science behind this. And then finally, the Simcoe Muskoka District Health Unit takes the lead typically on determining blue-green algal blooms, and they also sample swimming beaches for bacteria. So you won't see us trying to sample our bacteria near the swimming beaches. There's no need for that. All right, so now I want to shift gears a bit and take you through what the 2024 Water Quality Report has in it and what it says in a nutshell. The report is a members-only benefit and can be accessed once you've logged into the website. It isn't up there yet. It was a little late in getting produced this year, and there's still some edits to be made to it. So we hope to get that done and hopefully by the end of May. But we'll be putting out an announcement when it's available. So what's in it? Well, we have results from 54 sampling areas this year, or this past year, within 17 lakes and rivers, all done under the MLA program. There were 635 samples between May and September. In the report, you'll have summaries of the 2024 data in individual area reports for the Secchi dip, the spring and annual average total phosphorus, and the annual geometric mean of E. coli and total coliform bacteria counts. The results are compared with limits and plotted with long-term results, and the recommendations as well are in there in the report for where we might want to look at something a little different or analyses to undertake. So the consultant provides some advice to us on those things. So where we sample, there are 54 areas, 17 different lakes and rivers, deep water and near shore. And you can see on the map here that the dark blue are the deep water, the yellow are along the near shore. So you can see them congregated in certain areas, like down at in Gravenhurst and around Port Carling. The aqua color are the rivers, and the orange are areas of special study where we've had an issue and we've taken a few extra samples in areas to try and discern where the issue may be coming from. You can go on the MLA website and pull the map up and then put your cursor on a dot and it'll bring your area report up. So you don't have to go through the whole report to go one by one by one. So that's a very helpful thing to do. All right. So there are generally three kinds of lakes. Eutrophic, where phosphorus levels are 20 micrograms per liter or greater. And an example of that would be Lake Erie, which is usually considered a very productive lake, but also has had a lot of problems with oxygen and lagoon algae and fish kills, et cetera. There's mesotrophic lakes, which are between 10 and 20 micrograms per liter, so generally moderately enriched. And then oligotrophic. So those lakes are less than 10 micrograms per liter. They're nutrient limited, low productivity, and that's where the lakes of Muskoka largely are at. You can see that there's really not a big span between the three categories. You're talking about 20 micrograms per liter of difference between the three. So there's big implications when we shift from one into another category. So for what we found in 2024, almost half the phosphorus samples collected were below 5 micrograms per liter, which makes it very low productivity and very good quality. But we found 11% of the samples were over 20 micrograms per liter. So while you could consider them eutrophic, that's not the whole status of the lake, but it says they're quite enriched in the near shore, and that's where you may see algal blooms growing or a lot of plant growth. And then deep water phosphorus sample concentrations at all sampling areas were below the provincial water quality objective of 20 micrograms per liter to prevent nuisance algae, which is very nice to have that result. But in all but one, which is Brandy Lake, were below the provincial water quality objective against aesthetic deterioration, and that's 10 micrograms per liter. So basically all but one sample was in that below the 10 microgram per liter. So putting again into that oligotrophic category. And similarly, 90% of the nearshore results are also less than 10 micrograms per liter, and only 5 sites exceeded 10 and 2 exceeded 20. So overall, very good water quality in the lakes. This is an example of the summary charts you'll find in the report, and this one shows the variability amongst the sites. This is for all the what we call the affiliate lakes and the river sites. So you can see that the black is the deep water, the yellow is the near shore, and the blue are the rivers. And you can see the variability in the near shore much more than the deep water. And that's because they're taking all these little bits and pieces of things that come in on a seasonal basis or when it rains and assimilate them there. And then we have charts for each of the areas that show the long-term trends against the thresholds to see if things are getting better or worse or staying the same. And you can see, again, that there's been, and it's also showing you from 2002 on the left up to 2024 on the right. So you can see that we are seeing, I think we're seeing levels coming down for the most part, and probably that's because of policy that's been put in place around where we site septic systems and buffers and things like that over the years. And then this is an example of a chart depicting the long-term E. coli bacterial results from samples collected in 2024 as summarized in the area reports. This example is from Bala Bay, and in total, in all sites, the total coliform samples did not exceed the threshold at any site. There were elevated concentrations. Where the elevated concentrations were, though, were usually because they were after precipitation they were sampled or analyzed after precipitation events had happened. The E. coli accounts exceeded our limit of 30 at 18% of our sampling events, 36 events. So that's, again, not a bad result at all. But of the 36 exceedances, 75% were associated with precipitation events. So it's really important to try and sample during dry weather so that we're not seeing those kind of anomalous results just because of rain. And it just brings me to a reminder that it's really important to resample when you found the results to be high. And if you can't, please advise your lead or the office, as it might be possible for someone else to collect the sample. And I know it's hard because it could be, you're going back to the city for the week, and it would normally occur after the weekend sampling. So. And then this is an example of the area reports that show where the sites are located on the map, and the plotted with the results from the other years to show the long-term trends. And the results are also shown, the actual measured result is also shown. And Ariane, if you click again, on the second page of the result of the area report is a short analysis by our consultant with the light assigned to the site. There's the green light there saying East Bay is green light. All right, and then this year our consultant did a new evaluation for us, and they compared the results of the deep water sites with the nearshore sites to see if there were significant differences. So you can see what it's showing here, showing the 75th percentile of the results, the median, and the 25th percentile of the results, and then the lines represent I'll say outliers, but results that were outside those results, the 75th to 25th. And so what you can see on some of them, on the top one where the arrow is very consistent. Sampling the near shore isn't really giving us a lot more information because the deep shore is telling us what we need to know. And then some, like the second arrow, are showing us that they're all over the map. It's very widely scattered. So it makes sense if we only sampled the deep water, we wouldn't catch all the variability in those samples. So it will help us to decide if we need to reduce some sites. Once in a while, we want to add some sites, and we can't always add the budget. So we want to sample where we have the best results. We haven't had a lot of time to actually go over this too much yet. So we haven't reduced very many sites as a result of this, but that could happen next year. Sorry, okay, there we go. So water quality in our lakes ranges from this year from 5.8 meters at the appropriately named Clear Lake, and Lake Joseph is the clearest of the three big lakes, averaging this year 4.6 meters, which was a slight reduction from 4.9 meters we found last year. Followed by Lake Rosso at 3.25 meters, also a slight reduction from 3.5 meters averaged in 2023, and Lake Muskoka at 2.7, which is also a slight reduction over the 2.9 of 2023. And the lowest reported Secchi result was in Alport Bay on Lake Muskoka at 1.7 meters, putting it into a sort of a mesotrophic category. So. And the rest all stayed also said they were in the oligotrophic category. So the reduction in the clarity, I started to go, because Clear Lake was 6.8 and now was showing 5.8. Was this because we redid the ropes on the Secchi disc last year, and now you can actually read the results? But I'm seeing a lot of the sites showing a slight increase in phosphorus over 2023. So it's quite possible that that there is a little bit of decrease because of water quality. Hutchison did do an analysis of the trends on our problem, only on reporting on the problem areas, and found a small but significant decline in Secchi depth, which may be indicative of increased productivity at those stations. So something for us to keep an eye on and look for in our 2025 sampling. And then we keep track of confirmed occurrences of blue-green algae annually, and this means they have to be confirmed by either the Ministry of the Environment or Simcoe Muskoka District Health Unit. And this shows the lakes with confirmed algal bloom since 2020. As you can see, there's quite a variability over the year, and some more consistently show up like Leonard Lake or Silver Lake and 3 Mile. This year we did experience several blooms in new places, 2 around Bala in the Moon River, and at 3 of the affiliate lakes, Silver Lake in Township Muskoka Lakes, High Lake, and 3 Mile Lake. And then, as I mentioned earlier, new science coming to us from the Friends of Muskoka Watershed is showing impacts on freshwater Daphnia, which are their little zooplankton that eat the algae. Those effects seem to be as low as 10 milligrams per liter and some up to 50 milligrams per liter. So we've been sampling some locations on the lakes to get a sense of what it might look like for us. And the circles there from the red arrows show around Muskoka Sands having almost 20, and Muskoka Bay, Gravenhurst having 15, 16, even Hamer Bay and Lake Joseph, which it was a surprise, is at 11. So, and it's wide open. So it's kind of an interesting result. But so we've been adding a few more sites as we go. So please check your kits carefully for the locations. We're only doing 1 sample in May when the water column is mixed up, and it gives us as close to winter runoff as our program can get. Friends of Muskoka Watershed is out doing winter sampling as well. So this will hopefully help to start to create a picture of what's happening with salt in our watersheds. So as I showed you earlier, we're using a stoplight format to represent the parameter and if it meets the thresholds. Next. So we have been aligning our thresholds. Next one, Karana. Yeah. With the District of Muskoka's Lake Health Policy. And this year it's under review, but nothing has changed at this point. But next year that we may have to review our parameters. The MLA assigns a yellow light threshold for phosphorus concentrations that are over 20 micrograms per liter, or showing an increasing trend, or have an average annual total phosphorus that exceeds spring total phosphorus. Red light threshold for phosphorus is triggered if concentration exceeds 20, and if there's a statistically increasing trend in spring measurements over the last 5 years. In addition, a red light occurs for study areas where confirmed cyanobacteria or blue-green algae bloom is detected in the current monitoring year, and a yellow light is assigned if a bloom has been confirmed in the past 3 years. A green light for a green light can be returned to an area if there hasn't been a bloom in 3 years since the last one. And so this is what the results of the threshold analysis produced. Of the 54 areas sampled in 2024, 41 were assigned green lights, 13 areas were flagged, 8 were assigned yellow lights, and 5 were assigned red lights. All 8 yellow lights were the result of elevated bacterial concentrations. 4 of the red lights were assigned to Bala Bay, High Lake, Silver Lake, and 3 Mile Lake because of confirmed algal blooms. The 5th was assigned to Willow Beach, where we've been, that's our special study area where the yellow sample sites were. We've identified a long-term statistically significant trend in phosphorus concentrations, as well as elevated bacterial concentrations as well. And so overall, there's way more sites than we've had previously. This is an increase from 2023, where only 5 areas were flagged, 4 yellow lights, and they were all due to E. coli exceedances, and only 1 red light, which was also at Willow Beach. On a positive note, Windermere is no longer on this chart this year. So is that an anomaly, or is it going to stay that way? So we're looking forward to seeing what's happening this year. And the consultant has identified Muskoka River, Minette, East Portage Bay, Gull Lake, Hamer Bay, Clear Lake, Star Lake, Willow Beach, and Beaumaris as sites to watch. And they provided a few recommendations coming out of the report. And one is to continue to review the sampling stations so that we get the best data we can for the expenditure. And to have consistency in our data collection is vitally important. So continued training is important. So thank you all for continuing to make sure you're doing it as everyone else is. And as is dry weather sampling and retesting of high bacterial sites. So they're very insistent that we do the best we can on that. They want to consistently carry out more a detailed analysis of the data we have. You have a 20-year record now, so that's really important for us to mine it for these stories it's going to tell us and the insights it's going to bring us. And to continue to focus on our hotspots. And we will do a better job this year of trying to get that message out to the municipalities as well about what we're seeing. So that is it for the overview and the report. And I'm just going to ask Kariana to see if there's any questions in the Q&A that we might need to look at. I don't see any questions so far. But if you have them, for those of you who may join us late, the Q&A at the bottom of the screen on the right-hand side, you can put your questions in there and we can answer them either by written or we can verbally answer them as we go through. So we will come back and check this as we go. Oh, there's one. Why the comparison of the deep and near shore? I'll answer that verbally, is that we were looking to try and determine if we're using our resources the best way. If they're giving us the same result, then we don't need to have an additional site. For example, one of the sites down by Touchstone, the deep water site and the near shore site are giving us almost exactly the same answer. So why would we waste the money on sampling that? We can allocate that to another site where we're seeing greater variability. I hope that covers it for you. All right. Oh, here's another one. The question is, Lake Joseph, I'm told, turns over much slower than Rosso or Muskoka. Is this true? And if so, why? It is, as I understand it, it's true. I don't have the actual turnover rates, but partly because it doesn't have the same input. So as I was mentioning at the beginning, Lake Muskoka takes the 5,000 square kilometer watershed with the South Muskoka River through its lake. So it's going to turn over much more rapidly. And then Rosso has also a few smaller rivers entering in, and it also takes Lake Joe, so it's going to turn over that. And Lake Joe has very few inputs, and certainly nothing big, nothing like a large river coming in. So yes, and I think it's a little deeper as well, if memory serves, but. And then I'll take one more here. Oh, well, we're getting all sorts of them. They recently redid the roadway on Beaumaris Road, and it seems to push significantly more water into the lake from up the hill. I was there on a rainy day, big rain, and the dirt and debris was in the water past the docks. Oh, well, I can't answer that, Carolyn, but thank you for that information, because that may be also contributing to what we're seeing in our results. And I believe you're going to be over there helping to monitor for that. So let's keep that one on our radar screen. And Louise is listening, so she'll do that. And oh, there is 3 Mile Lake missing from the sampling map, and I'm sorry. Yes, it may be, and we will be including it. That some of those maps haven't been updated, and hence some other things we will be talking about as we get through the training part. So, all right, we're going to move on to the training. Thank you for the questions, and feel free to add more. We will come back to them before we end today. All right, next slide. Mariana. There we go. Oh, all right. All right, so we've got our training video and
Hey, I'm Jan Getz, and I'm one of the volunteers in the water sampling program. And today we're going to look at how you do your water sampling. First of all, we're going to talk about the equipment that you need and then what you need to do before you go out into the boat. Every team receives one of these bins, and it includes a binder, and a box, a bin that has a Seffi disc, a filter. You may have this type or this type, a thermometer, a collection bottle, potentially a bacteria sample bottle if that is on your requirements, or bottles for your phosphorus samples. You will also have a Sharpie that you use to put the dates on the phosphorus bottles, and a wax crayon that you use to put the site location on your bacteria bottles. In addition, you will need to have a cell phone, a cooler with ice, preferably ice cubes, and a life jacket. If you are missing any of these materials, and there is a list in your bin of what you should have, please let the office know to make sure that before you start to go in the boat, that you are prepared with all the material. You need to become very familiar with your manual. The protocol of the is outlined in it, and it does change from year to year. So please make sure that you are familiar, whether this is your first year for sampling or you've done it for 10, 15 years. The other things now that we're getting ready to go out is know what the schedule is. There's a schedule for when you go out and requirement as to what you're going to do each time you go out. First of all, you're going to fill out your data sheet, and it will have the date, the time, the names of the trained samplers, and anything else that's there. It's very useful before you go out to put what the locations you're going to and what you're going to be sampling for. In this case, this is the second week that we've been out. And so in this location, we're only doing a Secchi disc. But at location number 1, this is a 0, a deep water site. In location number 2, we're doing both phosphorus and bacteria. The other thing that you will notice, this is the sheet that goes with that clarify, so you know what it is. So this is the one, and as I said, in week 1, you did a phosphorus, but going through, you did a phosphorus and a bacteria, and that will continue on through. Be very careful. In many of the sites, there will be a duplicate in your 0 site on the first week. There's never bacteria on the first week because the water's too cold. You also will have a map that shows the location of your sites, plus picture visuals and the GPS coordinates. And those are very important in order that we have continuity from year to year. Then I think once we've got that done, we're ready to go on into the boat. Now Chris and Louise are going to demonstrate deep water sampling. They've located the site using the GPS coordinates in their manual and matched that with the GPS on their cell phones, or possibly you've got GPS on your boat. Note the water conditions and mark that on your sheet. Today it looks very flat, so this works well. Hang the thermometer put it into the water on the shady side of the boat. It's best to have it tied to the boat so it doesn't get lost. And remove your sunglasses if you've got them on, and then start lowering the Secchi dish into the water. You're letting it go down until you can no longer distinguish between the black and the white segments. Once you reach that depth, please mark it at the water line to the nearest 1/10 of a meter onto your data sheet. Then continue to lower it and then start to raise it again. That first measurement was Secchi down, and now as you're starting to raise it and you measure when you can again see the segments, that is your Secchi up measurement, again to 1/10 of a meter on the data sheet. And again, mark it with the clothespin. The distance between Secchi down and Secchi up should be less than 1/3 of a meter. Next step is going to be to rinse the collection bottle twice. Now is the challenging part of deep water sampling, and that's attaching the collection bottle to the Secchi dish. See that there are rubber ties and elastics, and now what we're going to do is lower the disc with the bottle attached down to the Secchi depth. You want to get water through the whole column, so you lower it slowly down until it's about between the two clothespins in depth. Should take maybe 3 or 4 seconds, but wait until there's no more bubbles coming out. Bring it up carefully. Dish. In the meantime, Louise is getting the sample bottle ready. No, first of all, she's going to get the filter and the sample bottle. The filter twice, the water that was in the collection bottle. And then run it through the filter into the collection bottle to the shoulder height. Don't overfill it. And you don't rinse the collection bottle because it does have special chemicals in it that we need. So be very careful when you're doing that. And of course, make sure it's the right bottle. Most of the deep water sites are 0. And then we're ready to move on to our next site. In this segment, Chris and Louise are going to demonstrate how to do a near shore sampling. They've checked their coordinates to make sure that they're in the correct location and have drifted into the location, making sure that the bottom was not disturbed. The goal is to get as close to the shore, about 4 feet of depth if possible, and it's easier to do in a small boat or in the front of a bow rider. First job, Louise is going to put the thermometer into the water, and Chris is going to check first that the sample bottle and the site number match, while Chris is plunging using a plunge and sweep method to fill the collection bottle. The plunge and sweep is put the water vertically upside down to about the forearm, turn 90 degrees, and sweep sideways. Once it's filled, sweep up. Make sure the bottle is full, the bubbles have stopped. He's now going to rinse the filter twice. And then using that water, they will fill the sample bottle up to the shoulder. And note, this bottle, which has material in it, is not rinsed. And be careful when you close it up to make sure it's tight so you don't spill it on the way back. If you're doing, Louise is going to put it in the cooler. This site requires a bacteria sampling, and so Chris is going to hope that his bottle is full enough that he can use that same grab for his bacteria sample. Again, the bottle carefully, the lid is carefully removed, making sure that either the top of the bottle or the lid are touched by your hands. Again, close the lid tightly, put it in the cooler. Now, we're going to retrieve the thermometer, record the temperature onto the data sheet, and note any abnormalities or things of interest. It could be construction taking place nearby, the presence of animals, or unusual vegetation growth. If you're concerned, you can leave this site after you've collected your sample, filled your sample bottle, and then do the sampling later. If your site only requires a bacteria sample, you can use the collection, the bacteria bottle on its own. It does not need to be rinsed. It's already been sterilized. You just use a plunge and sweep method. Put the lid on it and put it in the cooler. Okay, and we now return to shore, and in this segment, Louise is going to show us what we do with the bacteria samples. It is important, as we said, to have them in a cooler, and so it's necessary now to transform them from the cooler into a fridge until we're ready to return them to the NLA office. Now, at this point, Louise has set up the incubator, and it will be warming up to reach the desired temperature of 37 degrees Celsius. And it takes a while to do that, so it has to happen at any time. This is one of the reasons why it's important that there's coordination between all of the people collecting samples for each of the team leads, because they're the only people who actually do the incubation, and all of the samples have to get taken within 24 hours of the incubating. It's easier to both prep and read the results in daylight, so try and arrange that you can schedule it that way. It does take an hour for that incubator to heat up, and at the first time in the season, you want to make sure that you've tested it ahead of time. It might not have been quite right. The first step is to take out the number of color plates that you'll need for your testing run and label them with the site numbers. And you also need a blank color plate for the control test, which uses distilled water. And you need one of those for each incubator run. For each sample, after removing the lid, first of all, I think she has to go to the fridge and get the water, and then she is going to fill the collar plate. It's very important that you fill all wells of the collar plate. This is best done over a sink as it's messy. You do it with a back and forth motion for each set of rows, and then tap the collar plate gently to dislodge any bubbles. Make sure that all the wells are full by turning them in a good light to view, and then fill up any cells that are not full. Then you have to drain the excess liquid from the cells, the wells, by turning the collar plate so that the water runs off a corner. A paper towel at the edge can assist in this, but don't let it touch the wells. Then check that the wells are still full. The water surface appears concave in each cell. If there's too much water, then the wells will run together when heated and the results are impacted. Put a lid on the collar plate and the bacteria bottle. And don't throw the partially filled bacteria bottle out until all the collar plate fits safely in the incubator. You're going to repeat the procedure for each of your bacteria samples, and the blank, of course. And the blank is filled with distilled water. Now, it's best that the incubator is in an area with good circulation because it does have an odor. Some people have it on an outside covered porch if the weather is warm enough, otherwise inside. Put the color plates in the incubator, which we've done, and then on your data sheet, note the preparation date and time. Make sure that the thermometer is visible inside the incubator without opening the lid. Keep the lid closed, but verify that the temperature is correct. Now, you can rinse the bacteria bottles and let them dry and put them in your bin to return to the NLA at the end of the season. Then, 24 to 27 hours after the cold plates are incubated, 26 is best, you can get the results for the bacteria testing. So 24 to 27 hours after the cold plates are incubated, you can get the results from the bacteria testing. It's a two-step process. First, you're going to count the blue cells, which indicate the coliform count. Then you're going to count the cells that are blue and fluorescent, which indicates the E. coli count. For the first step, it's best if we put the colored plate on white paper to count the blue cells. You can count every cell that has any blue in it. Put that number on the data sheet under blue. To note that there are 96 cells on the color plate. Sometimes if the color plate is mostly blue, then you can count the cells that are still clear and subtract the count from 96.
Here is a poly plate that has, this one has all of the cells are blue. So I didn't have to do any counting because I know there's 96 cells, which is pretty scary. And this happens very rarely. This other one here, there's just a few blue down in the corner, but some of them are quite light blue. So this is how you can tell on white paper where they're light blue. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So I'll mark that down. But this is 96.
Okay. So now that that's been recorded, you want to take the cold plates into a dark room and put them on a dark surface or black paper. So we'll turn the lights out here and see how this works. There's a conflict between being able to film it and be dark. She's turning on an ultraviolet light and shining it on the colored plate so that she can count the fluorescent cells. Record the cells that are both fluorescent and blue under the data sheet where it says blue and fluorescent. And you can notice that those numbers are not going to be the same as the number of blue cells.
And you'll note on this one, none of them are fluorescent.
That's what we like to see.
Yes. The E. coli number is the most important number.
Now, if the blue and fluorescent count is 18 or higher, you will need to resample and should do 2 samples next time at the same location. Test a week later, so notify your team. You will likely need to rinse the bacteria sample bottles lids in very hot water and put lids on these for new samples. Occasionally, high readings are common and to be expected, which is why a retest is needed. If you are new to doing the testing, it's best to ask an experienced tester to assist you the first time, or you can watch them do theirs before you do yours. Hands-on experience is very helpful for this job.
Well, thank you to Jan and Louise and to Chris as well. I should have put him in as in our introduction as well. But thank you for that great video and so thorough. As you know, there are a few things that have changed along the way, but so we've got a couple of additional slides for you. But just looking at some of the reminders here that we need to have. Picking up and checking your bin. So picking up the bins, we should be able to be ready by Wednesday. We were waiting on some bottles to get in there and our summer student to start, which just happened on Monday. So check with the office before you make a trip in to pick up the bin. And if nothing else, we will have them on Seedling Day or Water Quality Training Day on the 17th of May at the Port Carling Community Center. They will all be there and available for pickup as well. And if you want one then and you might not be able to make it in the morning, let us know and we'll be able to hang around and wait for you because that is water sampling weekend. Confirm your team. We've assigned a number of new water quality samplers to several teams. Most of you have all made connection as far as I'm aware. But in case you haven't, please reach out to the team person or to the office again if you are not sure who you were supposed to have heard from. Please just remember to confirm your sites and parameters. We have made some changes. There were some inconsistencies amongst the various stations. I've tried to even those out a little bit, and I've been trying to make some room for some new samples in areas. So some of them I've pushed back to one sample just in the spring with Seki every time. So just make sure you've double checked what your assignment is for your sites, if you would, please. I'm going to try and reach out to everybody beforehand, but I haven't had the time yet, so we'll do our best. Avoid the rain at all if you can. Kariana's going to try and send out a weather alert beforehand so that we try to avoid significant rain. We had last June a significant rainstorm in the middle of our sampling weekend. We can't change the weekend because others have already gone out. So just make sure you try to do your best to avoid it if possible. It's difficult to interpret the results if somebody has a rain sample and other people don't. So it's trying to make sure we're trying to as consistent as we can amongst all the sites. Please take good and legible notes. What's happened as in the video, what's happening around you? Is it something look odd? Are you seeing something that could influence the results? And we really need to read them at the end because they're transcribed into a data sheet for future reference. Confirm and update the site coordinates and your pictures of your sites, if you would. Sites change over time. So we are trying to make sure our sites are all up to date, and our coordinates are all up to date. And that applies especially, I want to say, to the affiliates, because we've got some sites we don't have proper mapping for. So if you could all update the leads, particularly update your sites and pictures, we would really appreciate it. Just send it in to Kariana with your locations. If you can't do your weekend or retest, please tell the office. Maybe someone else can help. It's probably not going to happen, but just in case. And it does happen, that people aren't able to do their sites. It is part of being a volunteer organization. So thank you for being able to do it as often as you do. Next one, just flip past that. Okay. What we didn't have in the training video is chloride. So for those of you who are doing chloride, basically there's two different ways you're going to do it, is either through the deep water sample, there's a few of those, or a near shore. So basically you're collecting it the same way as the deep shore phosphorus sample, but you don't filter it. If you can if you have enough water left from your sample for the deep water coming up on the Seki disc, feel free to just use that for the chloride sample, or take another, do in the same way, grab another sample. And for the near shore sample, just grab and sweep through the shallow depth as you would do for a bacteria sample or for the phosphorus sample. No filter, cap it, and you're done. The BACT testing, as Louise was mentioning, it's sometimes it's difficult to see which ones are which in terms of the colored cells. We did get new BACT bottles this year. There was a little too much rust on the lids for our lichens, so there are plastic lids now, and all new bottles, all the same. We did have some issues with strange results. Did you have some of these results last year? These kind of like, is it a blue dot or not? So we had a few where that occurred. Um, we were in touch with the supplier, and they suggested that the pale colored results were as a result of spillage into other cells. That they were kind of diluted, and so that's why they resulted in that look. Um, they felt that it was potentially because they weren't drained properly, or when the lid got put on, that some of the water spilled into other cells, or that there was some condensation that resulted in the water also uh, being mixed up. So so the reminder is to spend at least a minute per Coley plate at a 90-degree angle to ensure that they drain off properly, and that then when you put the lid on, you're not spilling water into the other cells. Alternately, they suggested you could try not putting a lid on as so to reduce condensation on the Coley plate. Um, or we could use something called parafilm on the top if you want to have a lid on it. Cariana's got a little supply of parafilm, and that it allows gas exchange without condensation buildup or evaporation of the sample. So I think it's a reasonable solution. So Cariana has a supply if you want to try some, let Cariana know. We also tried filling the wells with pipettes, but for those of you like Louise who do a lot of those samples, it would be very time consuming and tedious. But for if you're only doing one or two sites, it might be the solution. And we are happy to get some pipettes if you're finding it an issue that is continuing this year. So just keep an eye, and we would want to hear about it, because if there's if it doesn't seem to be working, any of the solutions that the supplier has provided for us. And Cariana will provide you that stuff. So, and then just continue to remind, ideally retest if necessary. Just a reminder, last year we replaced the ropes on the Secchi discs. We did hear we had some bleeding of the red markered marks at the half-meter mark. I'm sorry about your white pants and your white boat seats. So hopefully there's sufficient mark left on the rope, but we would like feedback on their durability. If the marks are wearing off and they're not not able. We've checked the bins, but we weren't looking for every single mark on the ropes. They're all now in meters. A reminder, the black markers. The black marks is shown in the photograph here are every 10 centimeters, and the red mark is at a half a meter, and the meter mark is in red tape with a actual number written on it. So please let us know if they're not holding up, and if your ropes aren't holding up and they are fraying too much, or make sure the knots are all strongly tied before we throw them in the lake this year. So thank you for that. And then this is a very important note, is that the office has moved since you last dropped the samples at the door or into the fridge. So we are now located at 118 Medora Street, Port Carling, and that is just across from the parking lot entrance to the Turtle Jacks Plaza there. So the door is on the right-hand side of the building, but you'll come around the back. There's parking around the back. We're going to have our fridges out there. We're still trying to get the setup. Like, we just moved at the beginning of April, so I haven't had a lot of time to get this organized, but we're going to have a little setup for the fridges, preferably on some pedestals so that it's easier to get into them. We did have some broken bottles last year because they were jam-packed in there too much, so we're trying to manage that this year a little better. And there will be a bin outside beside the fridge for your field notes to be put in. And if you're in Port Carling and it's a weekday and the office open, go on in and say hi. All right, this is our sampling dates for the year, and you've all been provided with those. But remember, you can do it anytime from Thursday to Monday, and get your samples in so by Wednesday, so that they can get to the lab. And here's our contact information. You're welcome to reach out to either myself or to Cariana. Cariana is going to be the one that executes most of it, but I'm happy to hear issues that we need to fix. And again, thank you to all of you as our citizen scientists for your time and energy on this program. We have our environment committee that is very active in ensuring the program comes off every year, and especially to Cariana. And we have a new summer student, Beth, who are putting the kits together. And without them, we would be spending a lot more time in Port Carling. So thank you for that, everybody. Your kit will also have some flyers on membership for the MLA. So if anybody asks you who we are, what we're doing, and they're not a member, please feel free to inquire if they would like to become a member. And you have the a pamphlet there to provide someone if they're interested. And as always, your friends and neighbors should be members if they aren't. So please reach out to them and encourage them to take an interest in MLA water and Muskoka Lakes water quality. So thank you for attending. I will take some more questions now if anybody has them. And we will have the training videos are on the website today. And we will be putting up the presentation as well for you to refer back to in the event you need it. I'm just going to look at the questions here and see if there's any. There we go. See if there's any additional questions. I'm not seeing any. Oh, there's just a thank you. Thank you, Carol. Everybody's getting thirsty and hungry, I'm sure. My husband brought me a bottle, just a glass of wine while he was sitting here. So maybe someone's done that for you too. Just waiting to see if there's any questions coming in. All right. Not seeing any, but feel free if you think of something afterwards, feel free to reach out to myself or to Cariana, and we'll be happy to get back to you with the best answer we've got. So thank you for taking your time tonight. And thank you, Cariana, for getting us organized, and Jan and Louise for your support and star quality. I think we might need to export that to Hollywood. All right, see no questions. Thank you for coming, and we'll call it a good night.